Hello, this is Donna Mayer, Executive Director of the Cholangiocarcinoma Foundation. I'd like to welcome you today to the webinar. Today's topic is BAP1 mutations. The Cholangiocarcinoma Foundation is pleased to have funded a research study at MD Anderson to explore cholangiocarcinoma cases that have BAP1 mutations. We are very pleased to have today Dr. Rebecca Marcus and Dr. Milind Javle here with us to discuss their work to develop mouse models and identify better therapies for cholangiocarcinoma patients. Dr. Javle is a professor of GI medical oncology at the MD Anderson Cancer Center in Houston, Texas. He has also been a faculty member at the University of Texas since 2007. His research interests include identification of molecular subtypes of pancreatic and biliary cancers and development of novel therapeutics against these cancers. Thank you, Dr. Javle, and now turn the floor over to you.
Thank you, Donna, and the Cholangiocarcinoma Foundation for their support for our research. My talk is an introduction to BAP1 mutation in cholangiocarcinoma, and I will discuss the clinical aspects associated with this mutation, and then Dr. Marcus will discuss her fascinating work in mouse models. So I've organized this talk with an introduction to cholangiocarcinoma as well as the genetics of cancer. I will introduce BAP1 mutations, which is breast cancer associated protein 1 mutation, since these mutations were originally described in breast cancer. I will discuss the clinical implications of BAP1 mutations in cholangiocarcinoma as related to prognosis and therapy, and then briefly touch upon proposed research and the therapeutic aspects, including clinical trials. So cancer is largely considered a genetic disease. So genes, as we all know, are units of heredity that are transmitted from parent to offspring. And just to introduce this concept further, I want to talk about cell division, which is an integral component of growth and survival. As cells divide, that leads to growth and survival of all living species, including humans. And cell division occurs by DNA replication. So the DNA divides, and then subsequently the cell makes an identical copy of its genome or its genetic structure before it divides. And like any other mistake that can occur in nature, mistakes can occur during this process of cell division or DNA replication. And these mistakes are called mutations. And these mutations result in change of the genome in the new cell that is made. And these mistakes are usually repaired, but in cases these mistakes do not get repaired, then these mutations become deleterious, or these mutations become harmful. Now, mutations can occur in all kinds of genes, but if they occur in genes that control growth, or genes that normally suppress tumor formation, then you can have uncontrolled cell growth, sometimes leading to cancer. So the reason I give you this introduction is BAP1 is considered a tumor suppressor gene, and therefore a mistake in this gene or a mutation in this gene can lead to cancer. Now, these mutations actually take a long time before the cancer develops, and therefore cancer often occurs in the older age group. And it's often not one mutation that leads to cancer, but multiple mutations that act together that lead to cancer. Now, I also want to introduce this concept of germline versus somatic mutation. So the mutation can be in germline, that is transmissible from parent to child. For instance, there are certain breast cancers that occur in mothers and their daughters. Or the mutations can be somatic, and this is the large number, the maximum number of mutations that we see in cancers are somatic. They're seen in tumors only, and they do not get transmitted from parent to offspring. And I often get asked this question when a genetic profile is obtained on a patient is, what is my risk for transmitting this cancer to my child? And I tell them the difference between germline and somatic mutations And most mutations that occur in human cancers are not transmissible, or they're not germline mutations. So this is a cartoon that kind of explains the genesis of cancer from a mutation or a mistake that occurs in the DNA. So this mutation can cause the cell to grow abnormally and then eventually lead to tumor formation. This growing tumor can then invade the surrounding tissue, and then it can affect the blood and thereby spread to other parts of the body. So this sort of a simplistic way to imagine how a mutation in a single gene can eventually lead to a life-threatening cancer. So when we talk of cancers of the bile duct, we have to realize there are three different cancers. There's intrahepatic cholangiocarcinoma, or bile duct cancer that occurs in the liver, extrahepatic cholangiocarcinoma, that is cancer in the bile ducts that occurs outside the liver, and then gallbladder cancer, which is almost a completely different cancer, but also a cancer of the biliary system. These are three different cancers that have often a very distinct genetic profile. Now, clinically, when the patient is seen by the gastroenterologist or the oncologist or the surgeon, sometimes extrahepatic cholangiocarcinomas can be hard to distinguish from pancreas cancer. Intrahepatic tumors or cholangiocarcinomas that occur in the liver are often referred to as cancer of unknown primary, or cancers that occur from other that arise at other organs and spread to the liver, or non-biliary tract metastatic carcinomas. And the genetic profile of these cancers varies by type as well as region in the world. So this is the geography of the worldwide distribution of cholangiocarcinoma. As you can see, the predominance of cholangiocarcinoma is really in Asia, in China and Thailand. And often the kind of cancer that you see there, the cholangiocarcinoma, is different, not only clinically, but it can be different genetically from the cancer that you see in the Western world. So the goal of our group has been to study the genetics of cholangiocarcinoma so that we can identify novel biomarkers and targets that can be used to identify the disease biology and improve the outcome of this disease. We can then explore these targets for their therapeutic value by using novel inhibitors or targeted agents. And then we can use this information to identify some subgroups that might have variable outcome. For instance, certain genetic subgroups might have a better prognosis, while some might have worse prognosis. And this type of information is useful for planning therapy. But we do have problems when we do molecular profiling. So molecular profiling here I'm referring to is basically investigating the genetic makeup of the tumor. Often a tumor biopsy is obtained and sent for next generation sequencing, or to identify the various genes which are in the tumor that can be used for therapeutic benefit. When we investigate this platform of next generation sequencing for molecular profiling, unfortunately, we get adequate tissue for analysis in only about half the cases. And of the cases of the patients who we study, roughly about 20% of them have mutations that we can act on. So unfortunately, our knowledge of genetics is quite extensive, whereas the options we can offer in terms of therapies is far more limited by comparison. Furthermore, one of the disadvantages we face in the clinic is the turnaround time. When we send the tissue for molecular profiling, it often takes 4 to 6 weeks for the result to be back. So often the implications in terms of therapy and novel trials are not feasible in the first line setting. We often use them in the second line setting. So this graph explains the distribution of genes or alterations in about 400 cases that we studied with intrahepatic cholangiocarcinoma. So each of these vertical bars represents a different kind of genetic mutation. And the y-axis here shows the frequency of the mutation seen. So as you can see, there's some mutations are seen with a very high frequency. Unfortunately, the most frequent mutations are often not targetable. These include like KRAS mutation or P53 mutation, which are often not targetable. But less commonly seen mutations, for example, FGFR mutation or IDH1 or 2 mutations these days, have very promising investigational therapies. And this is the distribution of some of the more commonly occurring genetic alterations in biliary tract cancer. So what you see bolded here are mutations for which there are ongoing trials which appear to show preliminary benefit. For instance, the ERB2 or HER2 amplification that is seen in breast cancer is also seen in gallbladder cancer, and trastuzumab is a drug that appears to be beneficial in that setting. BRAF mutation, often seen in melanoma, occurs in a percentage of patients with intrahepatic cholangiocarcinoma. A very promising area is FGFR mutation, for which there are very promising targeted therapies being presented, and some data in this regard was presented at the GI ASCO meeting earlier this year and will be presented at the American Society of Clinical Oncology meeting this summer. IDH1 and 2 substitutions is another area where there are very promising targeted therapies. But the subject of the discussion today is the breast cancer-associated protein 1 mutation, or the BAP1 mutation, that occurs in about 15% of cholangiocarcinomas, the intrahepatic cholangiocarcinoma, and less frequently in extrahepatic and gallbladder cancer. And it appears to be related to a distinct clinical outcome. Before we go into BAP1 mutation, I want to discuss the value of molecular profiling and its implication on therapy. So for instance, in 10 to 15% of cholangiocarcinoma, we have now found there is a fusion of FGFR, where FGFR gene is fused with another gene, in this instance, called TAC3. There are specific inhibitors and to this mutation in clinical trials now, and this is, for instance, a patient in our practice who had multiple lung metastases. These are lung tumors that are seen on the pre-treatment film, and these with the inhibition with a specific targeted agent, there was a marked improvement in the lung tumors. There's a patient who had a BRAF mutation, a young woman who had very extensive disease. So this is a CAT scan that shows extensive cholangiocarcinoma involving the right lobe of the liver. And this patient was treated with a combination of dabrafenib and trametinib. And as you see in her first CAT scan, there's a very remarkable improvement in the liver tumor with this targeted therapy. Now, we found in case of BAP1 mutation that this mutation appears to have a relatively aggressive prognosis. So this is, for instance, a patient who had a tumor in the liver, in the left lobe of the liver. This tumor was then resected by a surgeon here. And within a few weeks of this resection, we found disease recurrence. We found spots in the liver, as well as, very characteristically, tumor that was spreading to the bones. Now, I have to say this is not the feature for all BAP1 mutant cholangiocarcinoma, but there's certainly a subtype of BAP1 mutant cholangiocarcinoma that appears to have a very rapid disease course, including spread to the bone with a higher predilection for bony metastases. So we recently, through the support of the Cholangiocarcinoma Foundation, we reviewed our experience with BAP1 mutation in cholangiocarcinoma, and this paper will be published within a few weeks, which describes the clinical outcome of this mutation, the patterns of spread, as well as survival associated with it. But going further into this mutation, I want to discuss in the next few slides as to what exactly is this mutation, what does it cause, and how can we possibly overcome it. So interestingly, BAP1 or the breast cancer associated protein 1 gene, was first discovered as a mutated gene in melanoma of the eye called as the uveal melanoma. And this paper that was published in Science in 2010 indicated that majority of patients having this melanoma in the eye, uveal melanoma, which is a rare tumor, have mutation in this BAP1 gene, and this mutation, this particular type of melanoma, in the presence of this mutation, was quite aggressive in behavior. But as with time, we have identified there are several other cancers that are associated with this mutation. Besides the uveal melanoma that I mentioned, it is also associated with an atypical skin tumor called epithelioid atypical Spitz tumor, skin melanoma as well, besides the eye melanoma, and a very peculiar tumor that occurs in the lung or the lining of the lung that I will discuss in the next slide, called mesothelioma. In fact, majority of patients with mesothelioma have this mutation. Now, I referred to somatic mutation, that is the kind of mutation that is not transmitted from the parent to the offspring, but found in the tumor alone. They have been described in mesothelioma, kidney cancer, cholangiocarcinoma, breast cancer, and more recently, colon cancer. Now, if the BAP1 mutation is confirmed in the tumor, we try to identify if there is any family history of one of these tumors, such as mesothelioma, particularly uveal melanoma. And then if that is present, then we discuss the possibility of a germline or an inheritable mutation, so that the patients can, so that we can then consider whether genetic counseling for the testing of the patient or the family for inheritable risk is appropriate. An interesting story that I want to describe in terms of this BAP1 mutation is that its association with mesothelioma was first described in Turkey, in Cappadocia. They found that this tumor, that in Cappadocia, Turkey, which is a very picturesque part of Turkey, by the mountainside, often the houses are built by the side of the mountains, and they found that the mountains and the rocks had a very high content of asbestos. It turns out that the most common cancer occurring in this population was mesothelioma, which is a very infrequent cancer. And when they tested this population in Cappadocia, Turkey, they found that majority of the patients with mesothelioma had germline or inheritable BAP1 mutation. So this was a good example of a germline mutation in association with an environmental risk. So it's sort of 2 + 2 = 10 here, led to an extremely high risk and high incidence of mesothelioma. In fact, at some point, it has been estimated that over 50% of the population in Cappadocia, Turkey, died of malignant mesothelioma. And this association taught us a lot about this particular mutation and its causation and its association with cancer. This again shows that while cancers may be different in different organs or different disease sites, the underlying genetic abnormality may be the same. So what does BAP1 actually do? So BAP1 is BRCA associated protein 1, and it is located on chromosome 3. It is on the short arm of chromosome 3. And if you look at the definition of what it does is it is defined as this family of D-ubiquitinases. So it's the proteins are bound to a specific ubiquitin moiety, and this ubiquitin is responsible for protein degradation. So D-ubiquitinases affect the binding of protein to ubiquitin, and I mentioned this to just to highlight the fact that the actual role of BAP1 gene is not known, but because of its impact on protein binding and its impact on protein degradation, it can regulate a number of cellular pathways that are including cell cycle, cell death, glucose production, DNA repair after damage. So how can we target BAP1? It is currently not known. It is known, the attempt is novel therapies, and it seems that a family of drugs called histone deacetylase inhibitors, maybe by, or HDAC inhibitors, such as vorinostat, may have value. In melanoma, particularly uveal melanoma, there is some interesting research with immunotherapy in BAP1 associated tumors. Currently, there's no data that BAP1 associated tumors respond any better to immunotherapy, but this is an area of research. So I want to highlight in the next few slides the role of novel therapeutics in BAP1 as well as novel therapeutics in cholangiocarcinoma in general. So novel therapeutics, often patients think of immunotherapy or genetically guided therapy, but actually it extends beyond that narrow definition. It includes new forms of chemotherapy, for instance, besides 5-FU, besides Gemza or cisplatin. We talk about targeted agents. Targeted agents are receptor tyrosine kinases or agents that actually act against the mutations that are active in that cancer. Certainly, immune therapeutics show promise in cholangiocarcinoma, although their role in BAP1 is less certain. And let's not forget there are other less targeted but still effective therapies, for instance, radiopharmaceuticals, such as Y90, radiation therapies, radiation sensitizers that have value in this setting, and their role remains to be investigated. Unfortunately, at the current time, there is a deficit of clinical trials in cholangiocarcinoma, and that's an area that we certainly wish to change. For instance, multiple myeloma, which is an unusual disease, higher incidence than cholangiocarcinoma, has probably 8 times the number of trials that cholangiocarcinoma has. Acute lymphoblastic leukemia, which is a disease that has probably similar incidences as cholangiocarcinoma, has almost 5 times the number of trials. So it's certainly an area that we need to work on. And that's the goal of a recently formed International Cholangiocarcinoma Research Network. This is formed through the sponsorship of the Cholangiocarcinoma Foundation. And our goal is to promote clinical and translational research and increase access to clinical trials for patients with cholangiocarcinoma, so including those with the BAP1 mutation. So I thank you, Donna and the Cholangiocarcinoma Foundation again for the opportunity for working in this area. And next, I'm going to request my colleague, Dr. Rebecca Marcus, to present some of her very fascinating work in regards to mouse models with BAP1 mutations.
Okay, thanks for the introduction. How do I make sure everybody can see my slides?
Oh, I think we're still seeing Dr. Javle's slides at this point.
Yeah.
While you're doing that, I actually will take this moment to ask Dr. Javle, there were a few questions for you.
Sure.
If you wouldn't mind. So the first one is intrahepatic cholangiocarcinoma or extrahepatic cholangiocarcinoma more common, and why?
So that's an interesting question, Donna, because the textbook teaching is that extrahepatic cholangiocarcinoma is more common than intrahepatic cholangiocarcinoma. It probably is still so, but the incidence of intrahepatic cholangiocarcinoma is increasing, and in many academic centers has superseded that of extrahepatic cholangiocarcinoma. That is artifactual. The intrahepatic cholangiocarcinoma is still classified in most tumor registries as primary liver cancer. So there's a large number of patients who are grouped in the liver cancer registries rather than as cholangiocarcinoma. And the second is extrahepatic cholangiocarcinoma has always been referred to as hilar cholangiocarcinoma, and sometimes distal cholangiocarcinoma may not be included in that definition. So it is a part of the problem of being a relatively uncommon cancer. This area has not received high priority in terms of reclassification, reorganization of registries. But to briefly answer that question, I would say that the incidence of intrahepatic and extrahepatic is reversing, and probably we are seeing a higher incidence of intrahepatic now. There are many reasons why that may be increasing. One could be that there is a overall increase in obesity and fatty liver, liver injury from substance abuse, such as alcohol or hepatitis. And some of these factors are contributing to an increasing incidence of intrahepatic cholangio.
Okay, the next question is if BAP1 stands for breast cancer associated protein, does that mean that people who have the BAP1 mutation have a higher risk or correlation with breast cancer?
No, they don't. Unfortunately, it's a misnomer, and its nomenclature was based on when it was first discovered in breast cancer. There is a risk in terms of familial or germline BAP1. A greater risk is of mesothelioma or of melanoma in the eye. There is a risk of breast cancer, but that risk is quite low.
Okay, and the last question is it possible to have more than one of the gene mutations that you showed in the chart early in the presentation? And it's a two-part question. And when it comes to a mutation, is the test measured as yes, you have it, no, you don't, or is it actually measured in the amount of BAP1 that you have, or the amount of that particular mutation?
So, well, that's very relevant questions. Most solid tumors, unlike liquid tumors like leukemia, most solid tumors are diseases of multiple mutations. So every tumor has several mutations, dozens, sometimes hundreds. But in this case, we look at the one that is most likely or most pathogenic, most likely to cause the cancer. We also look at mutations for which we have drugs, and we emphasize on that. Now, the second part of the question, remind me, Donna, what was it?
About whether or not, how it's measured. So is it measured in the amount of BAP1 that you have, or just you have it or you don't have it?
Yeah, so it's an evolving science. Currently, we get both types of information, that is whether the mutation is present or not. But we can also investigate further to ask for allele frequency. That is the frequency. That is how frequently is a mutation seen. We do not base our decisions, clinical decisions, on allele frequency at this point because it's not yet a validated measure. But I think clearly this is the way we're going to go in future, where we will not just look at the mutation, but at the frequency of the mutation, how often it is seen.
Okay, thank you so much for that. Dr. Marcus, it looks like you might be ready to go. I just wanted to share with the audience that Dr. Marcus is a clinical research fellow working under preeminent pancreatic cancer expert, Dr. Anabar Mitra. Dr. Marcus is an NIH T32 research fellow at the University of Texas and a general surgery resident at the Medical College of Wisconsin. Dr. Marcus, I now turn the floor over to you.
Thank you. And thank you to the Cholangiocarcinoma Foundation for having me, and for Dr. Javle for setting up what I'm going to talk about beautifully with his talk. So I'm going to be talking about the development of a mouse model for intrahepatic cholangiocarcinoma. And I'm going to break down this talk into 3 parts. So first, I'm going to give some background, go over a little bit of what Dr. Javle said and some other background. Then I'm going to give an example of a fairly successful mouse model that exists. And finally, I'm going to talk about mouse models in the context of intrahepatic cholangiocarcinoma and BAP1. So the background, we're going to talk about reviewing a little bit about what Dr. Javle had said for intrahepatic cholangiocarcinoma and BAP1. Then I'm going to talk generally how we study cancer and about mouse models and their role in studying cancer. The example I'm going to talk about is called the KPC mouse, and you will learn more about what that is when I get to that part of the talk. And then I'm going to end the talk by talking about existing mouse models for ICC and talk about the BAP1 mouse model that we are working on developing. So let's begin with some background. Very basic, cholangiocarcinoma is a primary liver cancer that develops from the bile ducts, and it arises from the cells lining the bile ducts, otherwise known as the epithelium. It can arise anywhere within the biliary tree, like Dr. Javle talked about. Specifically today, we're going to be focusing on intrahepatic cholangiocarcinoma. And again, this is within the liver. It does tend to be an aggressive disease that presents once it's locally advanced or even metastatic, and the therapies that we have can be limited for intrahepatic cholangiocarcinoma. And Dr. Javle showed that nice figure showing the incidence of cholangiocarcinoma across the world. It is more common in the Eastern world, but it has an increasing incidence in Western countries, specifically intrahepatic cholangiocarcinoma, not so much the extrahepatic cholangiocarcinoma. And in fact, over the last decade, we've really seen a marked increase in the rate of ICC in Western countries. And this figure shows that quite well. So starting at where the green arrow is and looking at the blue line, you can see kind of the sharp uptake in the incidence of ICC in the United States over the last 10 years. So the genetic changes that contribute to the development of ICC are not completely understood. Dr. Javle talked a lot about that, and that there are some mutations that have been found in ICC and genes that have been studied in other contexts before. These include genes such as KRAS and IDH that Dr. Javle mentioned, and BAP1, P53, and PBRM. And these fall into 2 broad categories of cancer-associated genes: oncogenes, which when mutated promote cancer, and tumor suppressor genes, which are genes that when functioning normally prevent the development of cancer, but when mutated or lost, then you can see the development of cancers. And the mutations that we've found in intrahepatic cholangiocarcinoma are unique as compared to those found in extrahepatic cholangiocarcinoma and gallbladder cancer. This figure shows that generally. So on the left-hand side of your screen, you can see several different genes that are involved in cancer development. And then the figure shows different kinds of mutations found in extrahepatic and intrahepatic cholangiocarcinoma. The colors correspond to the particular type of mutation, but you can see the colored areas are different for the 2 different types of cancer. Today, we're focusing on BAP1, which Dr. Javle did a very nice job describing in more detail than I did, but that I'm going to do. But basically, BAP1 encodes a protein, which is the D-ubiquitinase that he talked about, that's involved in DNA remodeling. And then as a result, it regulates many complex pathways that occur within a cell. And we know this from studying this protein in cells and in the context of other cancers such as the uveal melanoma and malignant mesothelioma that Dr. Javle talked about. So to discuss how we study cancer in general, if you can discover a tumor-specific target or something that's unique to the tumor, such as a mutation in BAP1, this can allow us to improve detection or diagnosis of the cancer and treat it at earlier stages, which ultimately leads to better outcomes. Cancer models help us discover these tumor-specific targets, and they really then enable us to have a preclinical model to study a cancer and develop treatments for these cancers prior to transitioning into the clinical setting. And there are two types of models. There's an in vitro model and an in vivo model. So an in vitro model is a cancer cell line that's derived either from an animal or from a human. And we study these cancer cells at the bench in a petri dish to understand better how different pathways within a cell work and are involved in the development of cancer. There are several advantages to the in vitro models. I just mentioned they help you discover the mechanisms that are occurring at the molecular level within the side of the cell. It's a very controlled condition because you're manipulating the cells within a petri dish at your bench, and so in theory the results should be reproducible. And also the cells should be relatively homogeneous, or they should be the same. The disadvantages to this type of system is that when cells adapt to living in a petri dish and not within the native animal or human that they came from, you could select for a population of cells that are have a particular feature that allows them to live in the petri dish, which may not represent what the cells are in the animal or human that they came from. They can also develop mutations the longer you grow them in the petri dish. Just as having the population that's very similar is an advantage in some ways for studying it. Also, there are many tumors are not just made up of one type of cell, and so that can be a limitation to this type of model. And finally, there's no TME, which stands for tumor microenvironment. So the cells are existing as their own entity. They're not interacting with any other tissue that's surrounding them, like would occur in an animal or a human. The other type of cancer model is an in vivo model, and these are animal models which mimic the natural history of human cancers, and as a result also mimic their clinical response to therapy. And they do this because they have mutations in them that are also found in humans with the same disease. And we'll talk about that a little bit more. So I like this figure because it just summarizes the way we study cancer. So this basic researcher at your benchtop in a petri dish, you're studying those cancer cell lines, that's in vitro work. And as you progress towards the clinical arena, you can have translational research or in vivo research in different animal models. And today we're focusing on the mouse. This is kind of the least expensive and most logistically manageable cancer model to use. So there are several types of mouse models that exist. I'm going to talk about three main categories briefly, and then we'll focus on the last one. So there's a chemically induced model of cancer, meaning that you have a mouse, you treat it with a carcinogen or something known to cause cancer. In this case, it's the DMBA. And then as a result, the mouse develops a cancer. This helps you study the traits of the cancer. It's a little bit more time consuming in the fact that you need to do some sort of sequencing or studying of the DNA to figure out what mutation happened to develop cancer after you treated with the carcinogen. Another type of mouse model is a xenograft, and in this case you take cells or tissue from an animal or a human and you transplant them into the mouse model, either under the skin or into the organ that the cancer naturally occurs in. And this is a relatively quick and inexpensive way to test potential therapeutic options for cancer. It's one of the most well-established ways that we pre-clinically test potential therapies that are coming down the pipeline. And the last type of mouse model is a genetically engineered mouse model, or GEMS, which is what I'll be focusing on for the remainder of the talk. So GEMS carry mutations in genes that are associated with specific human diseases, and therefore they mimic the disease. So, for example, we know that BAP1 can be mutated in intrahepatic cholangiocarcinoma, so a gem for BAP1 should have a mutation in BAP1. And in theory, then the mice would develop intrahepatic cholangiocarcinoma. These models develop a spectrum of changes so you can see the full development of the disease, or cancer in this case, from normal cells all the way to advanced or metastatic disease. And these models can be used in both an experimental and preclinical ways to evaluate different strategies to target your cancer of interest. And they're broadly classified as transgenic or endogenous. I'm not going to spend much time talking about this classification. Just suffice to say that the models we'll be talking about today are endogenous, and I will explain that as we get further into the talk. So moving on, an example of a successful GEM, the KPC mouse. So this mouse comes in the context of pancreatic cancer. So just very briefly, pancreatic cancer most commonly is pancreatic ductal adenocarcinoma, or PDAC. And it has a lot of things in common with intrahepatic cholangiocarcinoma. It's a very aggressive disease that tends to present at a locally advanced or metastatic stage. It has limited therapeutic options in some cases. And it's an increasingly common cause of cancer-associated death in the United States. The difference between ICC and PDAC is that for PDAC, several GEMs have been developed. The KPC mouse is a GEM that has expression of oncogenic KRAS and mutant p53. So KRAS that is promoting cancer, and p53 that normally would prevent cancer is now mutated, so you could get the development of cancer. And these mutations are known to occur in human PDAC, and in this mouse model, the mutations occur specifically within the cells of the pancreas. So going back to the basics of GEMs, I said they could be classified as transgenic or endogenous, and this GEM is an endogenous one. And this is how they work. So GEMs rely on the use of something called a site-specific recombinase, which is this, CRE. And basically what it allows you to do is target a mutation or a loss of a gene to a particular area of the genome. And so you take this specific recombinase and your gene of interest, which in this case are your KRAS and your p53, and within your target cell, which in the case of the KPC mouse is a pancreatic cell, you can get either complete loss of a gene, or you can get loss of part of a gene or mutation of part of the gene, whichever mutation you would want based on what you've seen in human cancer. So the KPC mouse is the most widely used of all pancreatic cancer GEMs, and it is genetically and histopathologically similar to human pancreas. So what does that mean? It's genetically similar. This is kind of similar to the figure I showed before, but we know that KRAS mutations and P53 mutations are common in pancreas cancer, and this mouse has both through the use of PRE. And it's histopathologically similar, meaning that these mice develop normal pancreases and then go through pre-cancer all the way to invasive cancer and finally metastatic cancer. So similar to what you would see in humans. And this mouse has been successfully used to study different strategies for targeting pancreas cancer, its detection, its prevention, and its treatment. And so finally, we're going to talk about intrahepatic cholangiocarcinoma, BAP1, and GEM. So the reason I talked about the KPC mouse is because pancreas cancer and intrahepatic cholangiocarcinoma do share many key features. For example, there is a multi-stage progression from normal tissue to invasive cancer. In pancreas, it's normal pancreas, and in cholangio, it's the normal bile duct that then develops into a cancer. Both types of cancers tend to have a very dense connective tissue surrounding it, or the tumor microenvironment that I referred to earlier, that it interacts with. Both of these malignancies often have early invasion into tissue outside of where it starts and distant spread. And finally, at least right now, detection of these diseases tends to be late, and sometimes that limits our treatment options. So given the similarities between PDAC and ICC, we would think that a GEM for ICC would be effective, given that PDAC has had such effective GEMs. So this is the figure that I showed before, talking about the mutations that we have discovered in ICC. The mouse models that currently exist for ICC have focused on these mutations, and we'll talk briefly about those. So the first mouse model that currently exists has an activating mutation in KRAS and a deletion of P53. So very, very similar to the KPC mouse, except that in this case, these mutations are targeted to the liver cells and not to the pancreas. And what results is that you get in the mouse a multi-stage progression of ICC with pre-malignant biliary or liver lesions, and you get the development of tumors surrounded by that rich tumor microenvironment. And so just like with the KPC mouse, you get a genetically and histopathologically faithful model, a model that produces cancers that look similar to what they look like in humans. And this figure just shows that. So this is showing how they use that recombinase PRE to induce the mutation in KRAS and delete P53. And these are actual figures from the mouse model showing the liver with large tumors that have developed. The second mouse model that exists is one that also has an activating mutation in KRAS, but it has a mutation in IDH2, which is another gene that's been found to be involved in intrahepatic cholangiocarcinoma. Again, similar to the first ICC GEM, it shows multi-stage progression with first liver cell expansion, and then you go into pre-malignant biliary lesions, and finally full-blown ICC. What's more helpful even for this model is that they used it to provide a link between the mutation and cancer, or in other words, to understand kind of how this mutation might lead to the development of cancer. And I'll show you how they did that generally. So this figure again just shows that these mice develop cancer similar to what we would see in humans, and this is a microscopic view of that cancer in this part. And the stains underneath, this stain stains biliary parts of the liver, which is where the tumor develops, and this CAR1 cell stains the other cells in the liver that are not involved in the tumor. And this is a somewhat complex figure, but I just wanted to show how a mouse model can really show this functional link. So they basically discover that if you have an IDH mutation, your liver cells no longer age and die like they would normally, but instead they continue to grow or replicate, and so you get expansion of the cells, the pre-malignant biliary lesions, and then intrahepatic cholangiocarcinoma. And that's what, with this model, they've been able to determine that this mutation promotes these cells that continue to grow and are susceptible to other mutations and then eventually cancer. So let's talk about BAP1 then. Currently, there is not a mouse model that incorporates a BAP1 mutation, even though studies have shown that BAP1 is often mutated in intrahepatic cholangiocarcinoma. So the goals of myself and the individuals that I'm working with has been to develop a new IHCCA gem that includes, again, that activating mutation of KRAS, as seen in all the other mouse models we talked about, and then a deletion of BAP1, which is a tumor suppressor gene. And just a reminder that BAP1 encodes that deubiquitinase, which is involved in DNA remodeling, and then lots of downstream pathways within the cell. So for my mouse, we again use that same site-specific recombinase CRE to target our genes of interest. And in this case, that's KRAS and BAP1. And in our target cell, that is the liver cells where cholangiocarcinoma would develop. Ideally, the cholangiocyte or the biliary tract. So when we did this, we have seen multi-stage progression of IHCCA, including the pre-malignant lesions. So this is genetically and histopathologically faithful model, just like the other IHCCA gems and the KPC mouse. And you can see that here. So these are pictures of livers from mice with the KRAS and mutation and the BAP1 deletion, and they develop lots of liver tumors. And at the microscopic level, these findings are consistent with intrahepatic cholangiocarcinoma. Now, as the second BAP1 model that I talked about earlier showed, they were able to explain the pathways and how the mutation in IDH2 helped lead to the development of intrahepatic cholangiocarcinoma. We are in the process of doing that with this new IHCCA gem. So in the future, we need to determine the functional link between BAP1 mutations and IHCCA, which Dr. Javle has talked about, that they're doing that as well. And at the same time, we will then be able to use this gem as a preclinical model to study strategies for detecting, preventing, and treating intrahepatic cholangiocarcinoma, with the ultimate goal being improved outcomes in patients and translation to the clinical setting. So just to review, this is what we talked about, a little bit more background and basics on how we study cancer in mouse models. The example of the KPC mouse for pancreatic cancer, which has been a successful gem, and the similarities between pancreas cancer and intrahepatic cholangiocarcinoma, and why we feel like a gem for intrahepatic cholangiocarcinoma would be successful. And then I talked about the two mouse models that currently exist for IHCCA, and the development of the BAP1 gem. and some conclusions. So IHCCA is a cancer of the bile tract. Currently limited treatment options because it often presents at a more advanced stage. We know that BAP1 is involved in IHCC, and that's involved in DNA remodeling and downstream pathways, but its exact role in intrahepatic cholangiocarcinoma is not fully understood. Genetically engineered mouse models, or GEMs, mimic human cancer, and so we can use them as preclinical models to study a disease of interest. And finally, that potentially a gem that incorporates a BAP1 mutation or loss of BAP1 may ultimately improve our understanding of intrahepatic cholangiocarcinoma and improve outcomes for patients who develop intrahepatic cholangiocarcinoma. And now I can take some questions if there are any.
Okay, yes, there's a few. You had mentioned pre-malignant lesions.
Yes.
Is this something that can be seen on a diagnostic test?
It can be seen histopathologically. So there are some lesions that you could see potentially on imaging, but they're often too small. And most of if you were to have an actual liver biopsy under the microscope, you could see those lesions, yes.
So at that point, would there be some therapeutic way to deal with the lesions, or would it just have to be kind of surveillance?
Sure. It depends on what lesion they see, I guess. There are some lesions that have been shown to be a precursor to intrahepatic cholangiocarcinoma, but can also be seen as a normal finding in the liver. And so in that case, if you saw that, you would just continue to observe. And then for the true biliary intraepithelial neoplasm, which are pre-malignant biliary changes, depending on which stage you saw in the biopsy, it would determine if you just observed or if you potentially did something.
Okay, thank you. We have another question about, are there any viral clinical trials for cholangiocarcinoma, and viral meaning viral attacking the liver?
I can take that, Donna. Did you say like viral therapy?
The question was, are there any viral clinical trials for cholangiocarcinoma? And then I asked, more specifically, and the answer was viral, like viral attacking the liver, like viruses attacking the liver.
Yes. So unfortunately, no, not to my knowledge. This is a great area of research and has shown a lot of promise for brain tumors, for instance. But it hasn't yet been attempted in a clinical trial in cholangiocarcinoma.
Okay, and then the last question we have is regarding the cholangiocarcinoma in cats and dogs. Is there anything that can be learned from studying the cholangiocarcinoma that they have, or is that different from human cholangiocarcinoma?
From that figure, I assume. So the cats and dogs are what are referred to as companion models. So they're just it's a spontaneous or naturally developing cancer that occurs in the animal that you can study because typically the time span is shorter in the development in those animals. I don't know if we've studied cholangiocarcinoma in the companion models. Cats and dogs are more prone to developing particular types of cancers, and as far as I know, they're not particularly prone to developing cholangio. But if they did, then yeah, we would study the native cancer in the animals and see what we could learn from that as well. It's different.
So Rebecca, I think it's a brilliant suggestion by the questioner. I think we should look and frankly, I haven't thought about it. Which animal species has an incidence of cholangiocarcinoma? that would be certainly an area where we can explore and investigate new therapies.
Well, actually, the questioner is me, and that is because I met a veterinarian at a recent conference that I was at, and she mentioned to me how many cases over her career of cholangiocarcinoma has seen and how prevalent she thought it was in cats. And then I happen to know someone personally whose dog has had cholangiocarcinoma, so that's what precipitated me asking that question.
So I think that's fascinating. We have to, I think, investigate, Rebecca, what is particular about this feline cholangiocarcinoma, and
Yeah.
one way to look at it is what kind of mutations those particular animals, those particular cholangiocarcinomas have, and if there is a particular type, for instance, BAP1, it would be a great area to investigate. So I was not aware of that, Donna. Thank you for that information.
Okay. Well, maybe that's another project that the foundation can fund. So we have one last, it's just a comment, and that is to thank both doctors for their wonderful work and their insight into cholangiocarcinoma. And of course, the foundation also shares those same sentiments that we truly appreciate all of your efforts in this field, particularly since it is such a rare disease. And we are going to end the webinar now. Thank you so much.